We are just waiting a few minutes for more attendees to join, and we will be starting shortly. Thank you for your patience. Well, good morning, good afternoon, good evening. Thank you for joining us today at this webinar. Um, Evidence at Your Fingertips, Safe in Pregnancy, Safe in Children, a Living Systematic Review Platform. I am pleased to welcome you on behalf of our group, which includes members of the Institute for Clinical Effectiveness and Health Policy in Argentina. This is a study or a work that is supported through CEPI and Speak and Brighton collaboration, as well as collaboration with Tulane and the University of Washington School of Pharmacy. And so on behalf of my colleagues, we welcome you to this webinar where we hope we can share with you our work regarding the Living Systematic Review platform. Next slide, please. Just as a little bit of a disclaimer, this webinar is going to be recorded and the recording will be distributed. You will find it available on our Speak CEPI website, and everyone who register will be able to receive a link to the recording, but you also can see it in our webpage. During the presentation, you will be muted if you are participating as an attendee. If you have questions, please go ahead and send them to us through the Q&A box, not the chat, but the Q&A box, please. Your questions will be addressed at the end of the presentation. And if you can, please identify yourselves if you would like to share your name and institution. And if we are not able to have a chance to address all the questions at the end, we will definitely make sure that the responses to your questions are provided separately at the end of the meeting. Next slide, please. So this is the agenda for today. I will give you a very brief introduction of the project. We will then discuss why we are working on a living systematic review, and this brief overview is going to be given by Agustin Chaponi. We'll have a demonstration of how to navigate the Safe in Pregnancy, Safe in Children Living Systematic Review tools by two of our colleagues, Mabel Berrueta and Agustina Mazzoni, as well as case by Agustin again. And then, as you see, we'll have Q&A and a discussion from your questions about the platform. Next slide, please. So I just wanted to bring you to the purpose of our work. As you know, CEPI, the Coalition for Epidemic Preparedness Innovations, has challenged everyone with a 100-day mission. And the 100-day mission is a global goal to develop safe, effective, and accessible vaccines, diagnostics, and also therapeutics within 100 days of identifying a new pandemic threat. And this is really the basis of developing a tool like the Living Systematic Review to be able to meet some of the challenges and the criteria here that are listed in these key pillars related to rapid information and ability to assess the safety of these vaccines, particularly in special populations. The next slide. is to give you again an overview of how the Safe in Pregnancy, Safe in Children Living Systematic Review really aligns with the CEPI 100-day mission. It does provide real-time and high quality evidence that is needed to include initially pregnant women was our goal, which relates to equity in rapid vaccine development. But you will see how this work has extended to other special populations, including children, those who have high risk conditions, and the immunocompromised. And you can see here the different areas of alignment of the Living Systematic Review with this 100-day mission, initially focusing on pregnant women, inclusion earlier in the clinical trials was one of the goals, but certainly equitable access. So looking at global data, including from low and middle income countries, the Living Systematic Review is going to help ensure that these groups, like pregnant women, pediatric populations, and other special groups, are not left behind when you have particularly a public health emergency with a new pathogen. We also provide an interactive hub, as you will see today, of real-time data that is going to be available related to the immunogenicity, effectiveness, efficacy, and safety of vaccines, supporting the 100-day mission global goal. And finally, the access to evolving evidence is really one of the highlights of the Living Systematic Review, which we will discuss today. Next slide, please. So to introduce you about the topic about why living systematic reviews, it is my pleasure to introduce you to Agustin Chaponi, who is director of the Argentine Cochrane Center and collaborator. Go ahead, Agustin.
Thank you, Flor. Well, I will present why living systematic reviews. Well, in this, I place the 100 mission, as Flor mentioned, in the center of this presentation, that is to accelerate vaccine development. So why living systematic review? First, to continuously update global evidence, real-time decision-making, to adaptive trial design and prioritization, to identify evidence gaps, for example, for prototype pathogens, for an early risk benefit assessment, and for global coordination and integration to reduce duplication and enhance collaboration. In one sentence, one evolving evidence source for rapid response. Next, please. I think that systematic reviews allow an objective analysis of the totality of the available evidence. But in the face of a changing landscape, only living systematic review can ensure that evidence remains current and actionable. Next, please. Next, okay. What is a living systematic review? Well, it is a systematic review that is continually updated, incorporating new relevant evidence as it becomes available. And why living systematic review? To timely connect evidence and practice through technological innovations such as online platforms, linked data, machine learning, as well as by broad collaborative research groups. The next, please. So when to perform a living systematic review? It's the intersection of three important domains. It deserves a LSR if there is high priority topic, significant uncertainty, and emerging evidence. So that is the exact point to conduct a LSR. Next, please. How we do this living systematic review? As in any systematic review and any research, we start with a protocol. In this case, we register as an evidence synthesis project. We register the protocol in Prospero. In this case, for example, Mpox, we start with two protocols, one for pregnant persons and another for children and adolescents, to ask the question safety, efficacy, and immunogenicity of mpox vaccines in these two populations. Next, please. And how is the workflow of a living systematic review? As any systematic review starts with a literature search and screening, we upload all the records in a software we are using now, Nested Knowledge, that is an AI-based software that use a robot that facilitate the screening and also a very good track of all the workflow. In a second step, once the studies are selected, we proceed with the data extraction. Pairs of reviewers independently extract relevant information in REDCap. That is a validated data collection system. That is a mini capture of the REDCap screen. And the next, please. The step 3 is data processing and quality control. We run periodically quality control scripts by a statistician, and then we proceed with generating databases and meta-analyses and summary of finding tables in HTML for easy visualization. And the last step is data synthesis and analysis, and also visualization. We use Power BI to present a dashboard, and also a Shiny app that is based in R program to provide customizable and lead real-time meta-analysis. And finally, with this evidence, we present in the grade. And the summary of this workflow is here. 1, literature research that is run every 2 weeks. 2, data extraction, that is double independent data extraction. 3, data processing and quality control. 4, data synthesis, analysis, and visualization that provide this interactive and customizable visualization, maps, forest plot, electronic summary of finding tables. And in the middle of this process, we place the artificial intelligence that is assisting us in several of these tasks and process. Next, please. Well, we started with a protocol and we finished with these mpox protocols with a recent publication in Drug Safety that is maternal and pediatric use of vaccine for mpox, a living systematic review and meta-analysis of safety and effectiveness. Next, please. Well, this is the final slide. So we place the AI and evidence synthesis, and we include the all the data process that is clinical study, literature research studies and literature search, data extraction, algorithms to select, machine learning, and then we proceed with the systematic review and rapid evidence synthesis, meta-analysis, and evidence summaries. And with the focus on the 100-day mission to provide safe and effective vaccines in 100 days, we also have the oversight, the supervision constantly of the humans, not only AI, but AI as a collaborator with the humans in all this challenging process. Thank you very much, and I will introduce now to Mabel Barrueta, that is Director of Department of Mother and Child Health Research in the IECS and also PI of this project. Mabel, please.
Thank you, Agustin, for the introduction. And hello, everyone. And thank you for joining today. I am going to walk you through how to use our platform powered by artificial intelligence, as Agustin explained, and more importantly, how it can support your work with up-to-date evidence of vaccines in pregnancy and childhood. While the initiative initially focused on pregnancy and pediatric population, it's now expanding to include the general populations, including adults. This platform is designed as a global public good, is freely accessible and continuously updated, providing high quality synthesized evidence on efficacy, effectiveness, safety, and immunogenicity for emerging vaccines. What makes this platform unique is that it's not static. Okay, the platform is structured around three main components. First, evidence synthesis. We systematically collect and analyze data from both randomized trials and observational studies. Second, this is some population search. Users can filter evidence by a specific disease, population, outcomes, key variables, allowing tailored subgroup analysis. And third, transparent outputs. All results are presented through interactive dashboard, forest plots, grade summaries, and another reports. The combination ensures that users not only access evidence, but can also understand and explore it easily by emerging vaccines, such as COVID-19, chikungunya, Lassa fever, Mpox, and RSV so far. So let me highlight some key features. We have a very smart search system where you can explore evidence by disease and refine it based on your needs. A curated database when all the studies are screened, tracked, and graded by experts. The platform has interactive dashboards which provide visual summaries of the evidence, and importantly, meta-analysis tools, including both pairwise and proportional meta-analysis. Finally, you can access research publications, including protocols, full reports. So overall, the platform is designed to move from data to synthesis to decision support. Now let's go step by step on how to use the platform. First, you select the disease or conditions, for example, Mpox. Then from the main menu, you choose what you want to explore. A dashboard, meta-analysis, summary of findings, protocols, prisma diagram, or quality assessment. These allow you to navigate based on your specific question. If you select the dashboard, you can explore different sections such as interactive maps, methodology, vaccine cards, quality assessment, and outcomes. And you can select different populations to focus on different populations you research. These dashboards provide a visual overview of the evidence, making it easier to identify patterns or key findings quickly. If you move to pairwise or proportional meta-analysis, you can customize your analysis. You can select the outcome, the population, the comparator, and the effect measure. This flexibility allows users to generate tailored analysis dependent on their needs. This is particularly useful for researchers, policymakers, and technical advisory groups who need specific evidence for decision making. The summary of finding tables provide a concise and structured summary of the evidence for each outcome. You will see a number of studies, total sample size, gray certainty level, and the relative effect. This is especially useful for quickly understanding how strong and reliable the evidence is. So now let's briefly discuss how to interpret the outputs. Very briefly, because then Agustina and Agustin will share some case studies just to use the platform and interpret the outputs. But the dashboards is to see the evidence evolves over time, identify gaps, detect emerging signals early. The gray tables, just to see certainty level, the direction of effect, and the number of studies by outcome and by population and by research question. And finally, the forest plots represent the, if it's possible, a pool estimate for an outcome just to answer my research question. In the three outputs, you can also click on results to go deeper into the original studies and reports. So to finalize, you can access by computer or cell phone. Our platform is not just a repository of evidence, it's an evolving evidence ecosystem that supports more informed decisions in global health. Thank you very much. I will introduce Agustina Mazzoni. Agustina Mazzoni is co-investigator of this initiative. She is a researcher of the Department of Mother and Child Health Research at Institute for Clinical Effectiveness and Health Policy. Go ahead, Agustina, and thank you for participating today.
Thank you very much. I'm very happy to be part of this webinar. Can you hear me well? Yeah.
Yes.
Okay, I will now present the first case study using safe in pregnancy to inform decisions on COVID-19 vaccines. Next slide, please. Safe in pregnancy was originally created in response to the COVID-19 pandemic when there was an urgent need for timely evidence on the safety of vaccines, especially for pregnant persons and children. This global crisis became the starting point for the development of the platform. Next, please. So, the first question we ask is, are COVID-19 vaccines safe during pregnancy? To answer this, we go to the Safe in Pregnancy platform, select COVID-19, and open the COVID-19 vaccine dashboard. This is where we start exploring the available evidence. Next, please. And the question becomes, is there evidence on the safety of COVID-19 vaccines during pregnancy? Do we actually have studies in pregnant persons? And the answer is yes. There are over 300 studies, including data from around 2 million vaccinated pregnant persons. These studies come from 50 different countries around the world, so the conclusion is we do have strong evidence on vaccine safety in pregnancy. Next, please. The next step is to understand how users can explore the evidence. The platform is designed to answer questions that users actually have about vaccine safety. For example, does COVID-19 vaccination during pregnancy increase the risk of stillbirth? Or does it affect the risk of congenital malformations? So the platform allows users to explore specific questions and quickly find the evidence. Next, please. Now we will show you how to navigate the website. If we want to answer questions related to safety, we select Outcomes, then we click on Stillbirth on the bar, and we can see that there are 57 studies reporting this outcome. If you want to see the meta-analysis for this outcome, we click on Pair with meta-analysis. Then, next, please, next slide. Then we select the analysis parameters. In the outcome type, we select pregnancy-related outcomes. In the specific outcome, we select stillbirth. In trimester of exposure, we select first, second, and third trimester. In the effect measure, we select risk ratio. And in the type of pregnant population, we select general pregnant population vaccinated. Next slide. Then we obtain the meta-analysis plot, which shows that vaccination does not increase the risk of stillbirth. Next, please. Then we can follow the same process to answer the other question related to congenital malformations. We click Outcomes, then we click on the bar of any congenital malformations, and we can see that we have 45 studies reporting this outcome. If we want to see the meta-analysis for this outcome, we click on pairwise meta-analysis. And then we follow the same process, selecting the analysis parameters. In this case, we select infant safety outcomes. On specific outcome, we select any congenital malformations. In trimester of exposure, we select first, second, and third. In the effect measure, we select risk ratio, and the type of pregnant population, we select again general pregnant population vaccinated. And then we obtain the meta-analysis plot, which shows that vaccination does not increase the risk of congenital malformations. This is my last slide. Thank you very much for participating. Now we go back to Agustin, who will present the chikungunya case study.
Thank you, Agustina. Well, the next, please. I present here some information worldwide about chikungunya. For example, in Europe, in October 2025, it was reported 700 cases, and in Italy, 353 cases. In some of these are still active points. And for example, in China, there are thousands falling ill with chikungunya. So that is a problem that is important, not only for Latin America or South America, but in the world. But under this important potential epidemic or pandemic, FDA announced that the safety of ICCHIC has concerns, so FDA suspend biological license, and US withdraw one of these chikungunya approval in August 2025. So the next, please. Our question can rise: are chick vaccines currently available under this suspension? Well, we can use our platform to answer this question. Let's click on chikungunya vaccine dashboard, and next, please. And then here, if you click in vaccine products, you can see all the clinical vaccine product details that we provide a vaccine cards table. The first of these is the ICCHIC that is currently suspended. And in this card, you have the manufacturer, the platform, live attenuated vaccine that not use adjuvants, the phase of development, that is phase 4, post-marketing, and when was the approval and the suspension. And at the end, we have all the clinicaltrials.gov documents and also all the publications that are related to this vaccine. The second card is an alternative. So the question and the answer to the question is we have alternatives. For example, the virus-like platform vaccine produced by Bavarian Nordic that is called Vincunia, that is in phase 3 development. And we have other ongoing vaccines, for example, an inactivated virus. And the next, please, you have another options that ongoing, that is live attenuated viral vectors and mRNA vaccines. So we are not, don't desperate, we have alternatives to ICCIC vaccine. Next, please. Another question, can you say, well, we have alternatives, but what is the safety profile of vaccine? Is there a one over the other that is best? For example, let's see one of the outcomes in the summary of finding table. For example, let's compare the arthralgia for vincunia, that is a virus-like particle vaccine, with the arthralgia produced by ICCIC live attenuated vaccine. Next, please. For adults, seronegatives. In adult seronegatives, vincunia produce a relative effect of 0.96, that is 28 persons vaccinated with vincunia per 1,000 have arthralgia, and 29 with placebo have reported also arthralgia in the clinical trials. So we didn't find any concern about this outcome. Unfortunately, the certainty of evidence was very low for this comparison that is provided by 4 studies. But one study provides evidence about the ICCIC vaccines, and there was a relative effect of 3.5 and 168 per 1,000 with ICCIC vaccines present arthralgia, and almost 50 persons per 1,000 produce arthralgia in the placebo groups. So that is provided high certainty of evidence. Let's see another outcome, that is fever, that is a common outcome. So if you see there is a relative effect of vincunia, almost two-folds, 1.8 more fever than placebo, but the absolute effect is not important, 16 per 1,000 and with vaccine, and 9 per 1,000 without vaccine. That is also provided by 4 studies. The certainty of evidence was low in this case due to imprecision, serious imprecision. And what happened with fever in the ICCIC vaccine? In this case, the relative effect is very important, 17 folds, respect the placebo group. And the absolute effect is 139 per 1,000 with ICCIC and only 8 per 1,000 with placebo. That is low certainty of evidence. So if you can click all the outcomes that are the main outcomes that we selected, and also we have other ways in the forest plot to explore different frequent and not frequent adverse event comparing different vaccines. But the electronic summary of finding tables are a very good summary that provides for different interventions or vaccines, the relative absolute effects, and also the difference, and also the certainty of evidence that we provide in our platform. Well, that's here I finished the case study with chikungunya, and thank you very much for inviting me.
So thank you very much, Agustin, and also Agustina and Mabel for your presentations. We hope that by attending this webinar, you have been able to get a general idea of how the Living Systematic Review works as a platform, as a tool, and that you will have a little bit of time for discussion today, but also the opportunity to take a look at the platform yourselves. As you can see, again, it started with supporting CEPI's 100-day mission, calling for safe, effective vaccines within 100 days of a threat being identified. You can see, again, as a brief summary, how the Living Systematic Review, which started as a Safe in Pregnancy, Safe in Children, has now expanded to a platform that allows us to review in real time any evidence that has been generated on these vaccines for emerging pathogens. It really gets us to meet the challenge posed by the 100 days because, as you've heard, we continuously update the evidence on safety of vaccines, not just in pregnancy and childhood, but in other at-risk populations, including adults and the immunocompromised. This requires a process which you have appreciated today, from the development of a protocol to the development of the platform itself, the integration of AI tools along with the human expertise that comes from our colleagues at IX within the Cochrane Center in Argentina, expertise in terms of topics and the content of the work related to vaccines, vaccine safety, different pathogens, and also the special populations that we have described. And as you see that data is generated, you can look at the meta-analyses that are available to you and update the results in a real-time basis. About every 2 weeks, these websites the site is updated. So this really allows us to have a near real-time evidence synthesis, as you can see, that can accelerate the path that we're looking at from an outbreak to informing vaccine development for those who are working on developing new vaccines for these pathogens. It also will help support recommendations of vaccines as they become available, and further, vaccine safety and effectiveness assessments after implementation is also there. We do publish our reviews on a periodic basis. And so, as you've seen, some examples for Mpox, we have also work on Lassa fever at this time that has been published, and we are very soon to submit a publication with the data that we have focusing on these special populations for the chikungunya vaccines, as you have seen. The next slide is just to really invite you to go to the Safe in Pregnancy, Safe in Children website. As you can see there, we have the pathogens that we have discussed today and potential for expanding into new pathogens, including disease X, one that might not have been identified quite yet, but the platform will be ready and nimble to adapt to a new outbreak or a new pathogen challenge. And you see there the QR code where you can link directly to the website right now. If you go to the next slide, and please, I would like to invite all of our panelists to open up their cameras and be able to address some of the questions that come up for discussion. I see that there are many colleagues attending today that are involved in vaccine safety that represent different organizations. And so please go ahead and ask your questions in the chat if you like. So one initial question that we have, and please, Agustina, and Andy, thank you so much, Andy, also for joining us today. If you can turn on your camera, the one question that I have initially, and then I'll pass it on, is regarding the plans. Okay, I think Andy has already addressed that. From Dr. Goldstein, are there plans to include pandemic influenza vaccines, for example, against H5N1 in the project? And so, Andy, why don't you go ahead and answer for everybody? Thank you.
I'd be delighted to. So, yeah, thank you, Dr. Goldstein, for your question. And at present, there are no plans to expand to influenza vaccines. However, as you've seen since the presentation with respect to COVID-19 vaccines, the project really has expanded beyond its original focus. And this is based on a combination of evolving priorities brought to the team's attention and funding availability. So I think my best guidance at this point is if this is of interest to please contact the group. And with that, I'm going to turn it back to you, Flora, in case you or others wish to add anything to that.
Thank you. Thank you so much, Andy. And yes, indeed, I think what we wanted to really convey is the fact that this platform is able to address different challenges, and certainly it does require substantial effort, and this is where the funding comes from. But it can do that. It can look at pandemic influenza. It could look at new potential pathogens and address any new ones, provided, again, we have a planned project to work on this together. So yes, it could do it, but it's not currently one of the pathogens that we have on the list. I have another question from Dr. Molina, Felipe Molina. And this is regarding the post-marketing safety surveillance. It might be along your line as well, Andy, but also for the rest of the group. Maybe also, Agustin, I'll tag you on this one. The question is, how do you deal with the results of disproportionality analysis in big pharmacovigilance databases? Do you include data from other type of analysis in pharmacovigilance, such as causal inference or cluster analysis? And then, I guess there's also a question regarding rare events that are reported as signals, and have we thought about including evidence on the mechanism as well? Which I think this is a little bit of a different question in terms of once you detect these cases, what are the next steps in terms to understanding maybe a causality or an association or a pathogenesis mechanism when it relates to vaccine safety. This is it's outside the scope, Dr. Molina, of this particular project. Certainly, generating the data and potentially looking at trends in safety data, we think is contributing to at least identifying potential signals. But the mechanistic part is going to be, outside the scope of this project. But I would like to have Agustin answer the other part of the question, please. Go ahead, Agustin.
Yes, I will also ask the other panelists to help me to this set of questions. I said regarding the study designs, we will not have limitations in terms of phases of studies. We include post-marketing, and also we don't have limitation about the study design of comparative studies. So we initially include, and then we analyze separately. Even we have separate analysis for adjusted and unadjusted effect measures. So that is important to say also that we try to cover all the pipeline of vaccine development. So we have diseases like Lassa fever that is in the initial phase of vaccine developments, and we publish, in fact, a study that we include ongoing studies, even experimental animal studies. So that is the initial phases of the research to assist developers. And we also have studies that include post-marketing, like RSV, that we focus on post-marketing studies. So we try to cover all the pipeline to attend and to assist the vaccine development. Well, that's all. Any other panelists that want to add something?
Yeah, very briefly. I think that was well said, Augustine, that the information from post-marketing studies is also considered and utilized. But I also want to add that there is also a large immunogenicity component to the living systematic review as well. It doesn't get exactly at the question of mechanisms, but it does provide the basis for understanding, proximate outcomes, if you will, that I think are quite useful for vaccine development. And with that, I see there's another question. If I'm going to turn it back to Flora unless others have something to add.
No, thank you for that. We can continue this conversation because I think there needs to be a good understanding of what the source of data is and how the analyses are built, because definitely depending on the question, the research question. So if we're looking at immunogenicity responses or we're looking at vaccine safety, you could look at clinical trials, so pre-authorization clinical trials, and post. And the same thing, efficacy in the clinical trials versus effectiveness in the post-authorization. So we try to encompass everything. And indeed, in the platform, you can select what specific outcomes. And Manu has a question here regarding the platform, does it allow to create forest plots independently for a given research question? And I think it would be helpful to explain, these forest plots that were shown by Agustina and Agustin, for example, maybe you can also give a little bit more information regarding what comparisons you can actually make so that it is based on the data that is published. It is based on the data that is available and not necessarily, creating comparisons that are not supported by the evidence, by the literature, right? So I don't know if Agustin, you would like to comment on that, please. Go ahead,
It is
Agustin.
correct, Flora. Nothing to add. It is based to answer specific research questions, not to another question, like a tool that can be used for other meta-analysis. It's for the diseases and vaccines that we have already prepared.
Yes, the analysis take into account the published data. Only the data that author publish. We don't manage raw data. So we don't manage the individual data and databases, for example, surveillance databases. So the meta-analysis, the objective of meta-analysis is just pool estimate new effect measures, synthesizing or summarizing the data that is published by other authors. But as Agustin said, we cannot mix data. We cannot compare in a meta-analysis data that previously were not compared by authors. I think that maybe just to clarify regarding the question to surveillance, we alert, but we alert based on published data. If the surveillance database report publish reports, we include the reports. We include FDA alerts, FDA reports, and analyzed and alert the community with this new information. But we don't analyze individual data of surveillance databases worldwide.
And also said that under a question, under our research question, you can make sub-questions. So you can customize your question and analyze subgroups. There are multiple filters by region, by populations, as was mentioned, and also by vaccine product and another potential sub-question. So you have some freedom, but under our research question.
Perfect. Thank you. And that's why I wanted also to point out to Agustina, because you presented nicely the PICO questions. I hope you can still hear me. You presented nicely the PICO questions. And so that was a very good example to see how to start. And certainly the answers to that will be in the literature. So maybe you can comment on PICO and your presentation, Agustina.
Yes, what I try to present is that you can select questions. In the case that I presented, they were related to safety, but you can also select another outcomes. For example, I show you the stillbirth in the maternal outcomes, but you can select. There's a long list of outcomes like abortion, miscarriage, PPH, postpartum hemorrhage, and you can select the outcome. You can select the trimester that you want to the woman was vaccinated. Maybe you're interested in see what happened, for example, regarding PPH, in specifically the subgroup of women who were vaccinated during the third trimester. You can choose that, and you can see the meta-analysis specifically for that. The same regarding the safety, for example, for other infant outcomes. And also you can see evidence regarding effectiveness of the vaccines if you choose that filter specifically. There's a lot of, if you enter to the web page, you can see that you can choose whatever your question is. You can customize the meta-analysis you want to see. I don't know if you were referring to that, Flora.
Perfect. That's exactly it. Thank you so much.
Thank you.
So I wanted to also highlight that, again, this information is here in this link. This webinar is being recorded and will be available to you at the CEPI Speak website as well. And you will be receiving it as well as a copy if you register to this meeting. But maybe, Mabel, if you can move forward a couple of slides after the Q&A slides. I just wanted to share with you that we also have other videos where you can see some of the tools that we have at hand. Are you able to move forward, Mabel? Just
Yes.
pass the slides after the Q&A. I just want to show.
Yeah, I don't know if I.
Yeah. So this is just, we won't show them
Yeah.
today, but I just wanted to alert you, that there's additional tools available to you. This is one about vaccine safety and effectiveness. And I think we have another one after that is just basically available to you to show more information. I wanted, before we close, we have about a few minutes. Thank you for looking them up. If any members of the panel today had final comments, additional, words of wisdom before we close.
Well, as a reflection, I want to say that it is to be ready and prepare for a potential pandemic or a disease X, for example. It is important to live all the muscle of a group that is trained and prepared also to generate updated, quickly evidence, living evidence. So we are thankful, we are grateful with the CEPI Speak because they support this initiative, and we hope that we can extend our initiative and project.
I would like to thank CEPI and Speak for supporting this initiative. Really, I thank you for participating in the webinar. Please use the tools and contact us if you need help just to answer any PICO question, any research questions with our platform. We are in continuous improvement of our tools. We want to expand to other emerging pathogens and to reach all the researchers and decision makers with this tool. So thank you for participating, and thank you, Speak and CEPI, to allow us to continue with this project.
Well, thank you. And one last question from Dr. Davis. Can you just say again the name of the AI tool that is utilized in the process?
We are using it now, nested knowledge, but it's not the only software that we use. Initially, we use Covidence, and now we are using nested knowledge, but we are not limited to nested knowledge. We can also consider different options.
Wonderful. Thank you
If
so
I
much.
can quickly add, in this era of misinformation and lack of information and hesitancy as it relates to vaccine, I think that now more than ever, a platform like this can help in many different ways, not only for vaccine developers and policymakers, but it's an objective source of being able at your fingertips, which is the name of the webinar today, to be able to really understand how often these vaccines have been used and what the evidence base is. So thank you, everyone.
Wonderful. Objective and at your fingertips. This would be our parting messages. And I thank everyone in the panel today, everyone for your outstanding work, and for the participants for joining us this afternoon or this morning. Thank you very much.
Thank you.
Bye.